going to pivot to our most emergent patient group and talk about newborn screening. So I'd like to invite Jennifer Taylor to come up.
I'm Jennifer Taylor. I work for a company called RTI, which is a research institute. And I just want to thank the organizers, first of all, for allowing me to come and present and talk about our experience in North Carolina as far as newborn screening. Before I get started and go into details, I just wanted to acknowledge my team. Newborn screening is a system, so it takes many collaborators that works on this project. I work for, again, RTI. It's a research institute, and the mission of this organization is improving the human health, which means we research everything. But our particular center, we focused on newborn screening, ethics, and disability. And if I can work this pointer. Oh, yes. Don Bailey is the PI, and he's also the center director, and I'm the project manager for this team, and I am actively being involved in all aspects of it. As I said, newborn screening is a system, so we have multiple collaborators that we work with. We work closely with Duke University, who does the sequencing of the ABCD1 gene. That's in Catherine's lab. And then also we have 3 collaborators, David Millington, who's been very involved in newborn screening as far as mass spectrometry, Sarah Young, and Deeksha Bali. They have been helping us with the laboratory aspects. At UNC, they do the follow-up for the patients that are identified. So we work closely with Cindy Powell, who's a geneticist there, and then Kristen Kleinard, who is the genetic counselor. And of course, we have to work with the State Laboratory of Public Health. We've developed a strong partnership with them. We actually have a contractual agreement with them. This is the lab that's located in Raleigh. And state laboratories is very challenging sometimes to bring new disorders on. With the Baby Saves Lives Reauthorization Act, NIH and our federal entities recognized there was a need to have these pilot studies and early adopters to start the newborn screening for these new conditions. So they came out with this contract, which we were awarded. It's called an Indefinite Deliverable Indefinite Quantity Contract. And the purpose of this was to implement pilot studies for new conditions added to this RUSP. So we received it for, so we've done multiple pilot studies with the state. We've done a pilot study with SCID, which was funded by the Centers for Disease Control and Prevention. We did one for MPS1. We've just completed one for XALD, and we're also doing one for spinal muscular atrophy. And we closely collaborate with the director of the lab, who's Scott Zimmerman, and the newborn screening manager, Sarah Beckloff. So the testing method we chose for the pilot was developed at the CDC also. It is an HPLC tandem mass spectrometry method. That's a negative ion mode, and we're looking, like the other newborn screening laboratories, at the C26 LPC marker as well as the C24 LPC marker. And the assay is fairly simple after you get it developed. What we do is we just punch a dried blood spot specimen into a 96-well plate. We add a solution that has an internal standard in it, incubate it for a little bit, and take the extract out, put it in a new plate, and then it's injected in a mass spectrometer. So before you start testing, what you need to do is you have to validate your assay to make sure it's working properly in your state before you do full population screening. So what we did is, excuse me, we tested 2,000 de-identified specimens as well as screen positive specimens from other states. And this would help us so we can say this is a screen positive versus this is a screen negative. So the blue dots are all of the de-identified specimens, and we also had some normal controls from the other states as well. So they're all located down here. And then the other are were the screen positives from the other state. We received samples from New York, Connecticut, and Georgia. So you can see here that most of the samples fell to the right of the red line. So that was what we set as our initial cutoff. And this is, so they all, this x-axis is C26 LBC and the y-axis is C24. So most of the specimens had highly elevated C26 and C24. So we set our cutoff here, and then we set I'm sorry, I'm having trouble with this. And then we set our C24 cutoff here. So we noticed that there was some specimens too that just had an elevation in C24. So what we did is we set up a borderline C26, and then to try to capture those that had borderline C26 but elevated C24 results. So this is our screening algorithm. This is how it works in the lab. So we punch that initial specimen, we test it. And we look at both the C24 and C26 values. If they're normal, then it's a screen negative and nothing else is done. If it's abnormal, then we repeat it because we want to confirm the result. So we test it in duplicate, and we look at the 3 results that we have now. If it's in this borderline range, then what we do is we request an additional specimen. If it's in the abnormal range, then that's considered a screen positive. And what we do is we refer that child to follow-up, as well as we also take that blood spot specimen and do sequencing on it. So we send it out to Duke and we do sequencing on it. So here are our cutoff values. Again, the C26 was our primary marker that we looked at, and C24 was the secondary marker. So we had 2 ranges that were considered abnormal. The first was if you have a very high C26, so greater than or equal to 0.15. And then our second one is if it's in that borderline range, which was between 0.08 and 0.15. And then your C24 was significantly elevated, so that was above 0.175. So those that we thought were at highest risk for XALD. The borderline range is when we had just the borderline range of the C26. So what we did with those is we requested a repeat specimen, and then if both of them were low, if the C26 was low, then we said that was within spectrum limits, and those children were not followed. So the next thing we worked on is developing a follow-up diagram, and we worked very closely with the geneticist and the genetic counselor at UNC. So once a screen positive, a child is identified as a screen positive, there's 2 pathways that are taken. The first is the genetic counselor. They contact the primary care physician and ask if they want to talk to the patient, or if the genetic counselor, you want the genetic counselor to contact them. What then happens is the primary care provider is given instructions. So they're asking the families to come into their office, and then they would do an initial examination, and then also collect a specimen for confirmatory testing, as well as another dried blood spot specimen for the lab. So this was a challenge for us at first because a lot of the pediatric offices in our state are not used to collecting specimens from children. So some of the time they had to repeat the specimen. Sometimes they had to send them to another facility to get that specimen collected. The second part of this diagram was the sequencing part, which our team thought it was important to do this in the newborn screening program because our state has a hard time covering genetic testing for infants. So we thought it would be better that we do this in a newborn screening than after when they're in clinical care. So what we did is we took some punches from the blood spot specimen and sent them to the Duke Molecular Diagnostic Laboratory, and they sequenced the ABCD1 gene. So the confirmatory testing results and the sequence results went back to the geneticists at UNC, and then they could contact the families with further information. So if the very long chain fatty acids were normal and there was no variance, that was a false positive. We notified that family that this was a false positive and there's no other follow-up required. If the very long chain fatty acids were elevated, then we looked to see if there was mutations present. So if it was a male and they had a pathogenic or a variant of unknown significance, then that patient was seen in the genetics clinic at UNC. That for our families, that was a ALD diagnosis for them. And then there would be follow-up appointments with the endocrinologist and the pediatric neurologist. If it was a female that had a mutation, then they were also invited to come to UNC and get genetic counseling. If it was no mutations, then they also went to the clinic. In our case, it was either a Zellweger or other peroxisomal disorder, but they would do additional testing to see what disorder it was. So here's the screening results. We started screening at the beginning of the year. We started March 5th, and we screened all specimens that were received in the lab between January 2nd and June 1st. So we screened a total of over 50,000 specimens. We had 45 specimens that had borderline results. So in this case, we requested an additional specimen. And we received 27 back. They all tested normal, so no further follow-up was done. We did have 12 abnormal results, 3 males that had mutations in the ABCD1 gene, 3 heterozygous females, and then 6 referrals that had no mutations. 3 of those came back as false positives. So more about the screening results. So we had 3 males again. When they came into UNC for genetic counseling and initial evaluation. They had normal evaluations. There was no family history. 2 of the families had another child. The children had another brother that was around the same age. They had an examination which was normal, and they also got confirmatory testing done too. While the information I have on those other children is 1 came back with normal results, and the other one, the results were pending, so I'm not sure what the results were for that child. 2 of the moms decided to get carrier testing. 1 of the moms was a carrier, the other one was not, so the child had a de novo mutation. The heterozygous females, they were contacted, they received genetic counseling, but the parents decided that they didn't want to pursue any further testing at that time. They opted not to. So the approximate incidence in our population for the pilot was 1 in 8,724 births. It was an even distribution between males and females. The other screen positives that we had that had no mutation, so we had 1 Zellweger case. That infant was very sick at the time and was in the NICU. So the examination for that is they had hypotonia, developmental delays, they had feeding problems, they had abnormal eye movement, they had failure to thrive. And after they received those screening results, they sent out for a genetic testing panel, and it came back positive with a mutation in the PEX1 gene. We also did find a Cartier-Gutierrez syndrome. That infant was also very sick at the time and was in the NICU, had a very abnormal examination. We actually weren't able to complete confirmatory testing for that child because it was so sick and had so many tests done at the time, but they did do a genetic panel and it came back positive with a TREX1 gene. 1 case is still kind of pending. It might be a liver dysfunction. That child was seen at UNC and had a normal examination, but when we did confirmatory testing, had a slight elevation in the C24, C26, 22 ratio. So that child is going to be monitored for another year to see if that elevation goes down. And this is the same graph that I showed for the validation of where our results for the pilot study was. So instead of plotting all our screening results, I plotted the average for each day. So what was the average number, C26 number, C24 number for that day? So all of them were very, very low. They're in this blue the blue dots here. The orange dots are our borderline specimens, which falls in our borderline cutoff. The Zellwegers and the ALD case were very easy to find because both their C24 and C26 was extremely elevated. We had 3 heterozygous females. All three of them had elevated C26 values, and 2 of them had elevated C24s. What was tricky was the ones that had elevations with the C24 and not the C26. So the 2 false positives had elevations in C24 and not C26. There was one case that came back borderline twice, so we wanted to just make sure that infant was negative. So we did send it out for confirmatory testing, and it was negative, so it was false positive. The one case down here is the child that had the Cartier-Gutierrez disease, and then the other case that we found up here, the pending possible liver dysfunction. So here's the confirmatory testing results. We had very good, most of the confirmatory testing results were done at the Kennedy Krieger Institute. And we had great correlation between our screening results and the confirmatory testing results. The only there was one infant that's not on here, that was the one, it was very sick at the time, so they sent a sample to LabCorp. That was a problem in our study, is educating pediatricians where to send their specimens to. And it was a bad sample, so it had to be repeated. Here's the sequence results for our study for all the infants that were identified. Most of the ALD kids, they had a VUS. We had to classify it as a variant of unknown significance, but when we looked in the database, there was a pathogenic mutation at that same position, but it was a different amino acid change. So we called it possibly pathogenic, but it was classified as a variant of unknown significance. We did have one child that we thought was a peroxisomal disorder at the time, because it did not come back with any mutations on the initial panel. And after it had confirmatory testings that was also elevated. So they did another genetic panel, and it turns out that there was a heterozygous deletion in exon 7 through 10, so which is not caught initially at sequencing. So that came back. Then we finally were able to diagnose that child as a carrier. So this is just this is information on where XALD newborn screening stands in the state, as well as a little bit of information about the newborn screening program in North Carolina. So North Carolina, there's approximately 120,000 newborns that are born every year. That number seems to be going up. More people are migrating south. They screen for over 31 core conditions, and they identify over 200 infants each year with these disorders. So the program exists on a budget that's around $3 million per year. Traditionally, the newborns, there needed to be legislative action to change the newborn screening fee and add new conditions. However, recently this year, actually this summer, they changed, they changed the process. So the legislators were tired of adding new disorders. So what they did is they increased the fee significantly, and then they added this acquisition fund, so they're hoping to make it easier for newborn screening to add new conditions, so you can purchase new equipment. However, the funding is somewhat limited because it provides a lot of funding to the lab, but the main problem with this bill is it didn't provide any funding to the follow-up program, which is also essential for newborn screening programs. And then it changed the process of adding new conditions in the future. So it added XALD, MPS1, and Pompe to the newborn screening panel, but then what they said, instead of legislators can increase the fee, what we're going to do is give it to the secretary, the HHS secretary, and the Public Health Commission, and they will evaluate the disorder to see if it will be added to the panel. So, So they're not screening yet. It's added to the panel. They're currently in the planning processes, and hopefully they will start screening soon. Another point I just wanted to make on this slide is long-term follow-up. So long-term follow-up has been a challenge with all newborn screening disorders, but it's particularly a challenge for XALD. What we did in the pilot is we were able to consent the families that screen positive so we can gather information for them. The funding source really wanted us to share the information with the longitudinal pediatric data resource, so we consented them for that. However, they did not provide any additional fundings for that. So currently North Carolina does not do long-term follow-up for any of the newborn screening disorders because they don't have any funding to do it. And when we applied to do this pilot, NIH did not provide funding for us to do it as well. But we are interested in that. RTI is very interested in contacting the families that we identified. So we are planning a study currently to interview these families that were confirmed with this XALD diagnosis and evaluate their access to care, how are they coping with this disorder, since none of the children we identified yet are symptomatic. Another point I wanted to make that it was a challenge for us when we were planning this pilot was cascade testing. So some programs are able to do this and test any of their family members that screen positive as well. However, we were worried about this because this could also potentially cause our program to go broke. So, but in our particular case, most of the families did not have access. They did not have a family history, so we did not do much additional family testing. However, I just wanted to point out it was an additional cost. So some of the moms did opt to get carry testing done, which was a cost out of their insurance pocket. We made it easier with having sequencing done so they could have targeted sequencing, which is cheaper than having sequencing of the whole gene. And this also requires a lot of workup and explanation if they do carrier testing as well. So I just wanted to thank you again for allowing me to present, and if you have any questions, I can answer them.